Assalamualaikum and good morning, doctor. We from group 5 will present video lab report calculations of hematocrit in fish. Before we start our presentation, I want to introduce my member in group 5. Number 1 is me, Elena binti Jinuddin. Number 2 is Siti Norada binti Isnil. Number 3 is Daniel Phillips. Number 4 is Onicha Evi. Number 5 is Maswe Martin. And number 6 is Christina Libia Suntai Anak Emai.
Hello, my name is Maswe Martin, and I will be showing you the introduction to the calculation of hematocrit in fish. According to the National Institute of Health (NIH), hematocrit (HCT) is a method to measure the volume of packed red blood cell or RBC relative to the whole blood. Hemoglobin (Hb) is a protein contained in red blood cells that is responsible for delivery of oxygen to the tissues. To ensure adequate tissue oxygenation, a sufficient hemoglobin level must be maintained. The amount of hemoglobin in whole blood is expressed in grams per deciliter. There are two common methods to sampling blood in fishes. First is acute sampling, involves the puncture of either a blood vessel or the heart, or the second one, the severance of the tail to drain blood from cut vessel. The second one, acute sampling, is common in all the studies, fieldwork, or whenever fish are too small for cannulation. Fish may be anesthetized, and or killed with a blow to the head prior to acute sampling, according to Gallagher and Farrell, 1998. Hematocrit levels in fish are a crucial indicator of physiological adaptation and response to environmental changes, diseases, and handling stress. Deviations from normal hematocrit values may suggest conditions such as anemia, parasitic infestation, or exposure to toxins, according to Nogat 2010. Thus, calculation of hematocrit in fish are proven useful in checking for conditions such as anemia, parasitic infestation, or exposure to toxin in fish. Thank you very much for listening. That's all from me.
The objective for this lab is to able to extract blood sample from fish, and second, to determine the proportion of red blood cell RBCs in relation to the total blood volume.
We move on to the materials for this lab. We have 10 different materials that we use. Firstly, we have the live fish. Next, we have the anesthetic solution. We use Transmo. We have the sterile syringes. We have the sterile needles. We have the anticoagulant, which is the EDTA, and the blood collection tube, which contains the EDTA. We have the capillary tubes, the centrifuge, the hematocrit reader, and finally we have disposable gloves and lab coats.
Hello, I'm Siti Norada binti Isnil, SQ2160351. Today I will present about method hematocrit. The first is blood collection method, fish handling and anesthetization. We followed appropriate protocols for handling and anesthetizing the fish. The fish were placed in a container with a suitable concentration of the anesthetic agent and allowed sufficient time to become immobilized. The third is blood collection. Once the fish were adequately anesthetized, were carefully collected blood sample from a suitable blood vessel, such as the caudal vein or dorsal aorta, while avoiding exercise, trauma, or damage to the fish. For hematocrit HT method, we examine the hematocrit value using the microhematocrit method. Heparin microhematocrit capillary tube were inserted into the blood sample until they fill approximately 3 or 4 of the tube. One end of each capillary tube was then sealed with a wax plug. The tube was centrifuged for 5 minutes at a speed of 1,500 rpm using a microhematocrit centrifuge. The hematocrit value was determined by measuring the percentage of erythrocyte volume from the blood using a ruler and expressed as a percentage HT.
My name is Sonisha Ivimekis, BS2216053. In this slide, I'm gonna talk about our lab result, which in the slide you can see 3 different pictures, which on the very left shows the first picture of a hematocrit that was divided into plasma and red blood cell after centrifuge for 5 minutes. By this, we can calculate the percentage of red blood cells from the total height of HCT or hematocrit. And the next picture, which is in the center, shows a sample of hematocrit. One shows a total height of 3.6 cm with the total length of red blood cell is 0.9 cm. The next picture shows the sample 2 of hematocrit, which the total length of HCT is 4.1 cm with the length of RBC or red blood cell is 1.1 cm. The next slide shows a table of raw data of 2 samples of hematocrit. Based on the table shows above shows that the percentage of red blood cell or RBC and the remaining plasma in the sample of fish blood that was taken. The RBC percentage in the sample 1 is 25%. Meanwhile, the percentage of the plasma is 75%. In the next sample, which is the sample 2, shows slightly higher percentage of red blood cell, which is 26.83%, and the plasma is 73.17%. That's all from me.
Assalamualaikum and good morning, doctor. I'm Elena binti Jamil BS2216005 and 08. Today I'm going to conclude lab report 3. In our conclusions, the hematocrit lab in fish involved measuring the proportions of red blood cells in their blood, which provide critical insight into their health and physiologically status. By assessing hematocrit level, we can evaluate the oxygen carry capacity of the blood, detect anemia or dehydrations, and monitoring the overall well-being of fish in aquaculture system. This lab is essential for ensuring optimal fish health, diagnosis potential issue, and implementing effective management practical in aquaculture operations. That is for me. Thank you.